the cell. The genes, the DNA, remember, we got that from mom and dad. That's all we got from mom and dad, right? They didn't give us all the proteins we needed. They didn't give us all the tools we needed. They gave us the information to make all the tools that we needed to grow up into the people we've become. Remember, proteins have a very special shape or structure that's determined by the order of the amino acids. Okay, so depending on the order of the amino acids, you get different shapes of tools. And then remember, proteins that are called enzymes make reactions happen. They speed up reactions by lowering the activation energy. Remember this graph? We'll just jot this down. Maybe this will be a good trigger for us. Right? Without an enzyme, with an enzyme. Wow, my computer died. Are you guys even on the meet anymore? Jen, are you still there? My computer completely kaputted.
We're here.
Okay, awesome. Sweet. Okay, so in order for us to talk about how we go from genes to proteins, we have to talk a little bit about another nucleic acid. Okay, we talked about DNA before. Deoxyribonucleic acid is what DNA stands for. Now we're going to talk about RNA, which is another nucleic acid. Okay, it stands for ribonucleic acid. Oops, somebody wants to come in. It's Brandon. Ribonucleic acid. And so that's what RNA stands for. R for ribo, N for nucleic, A for acid. It's another nucleic acid. It's actually very similar to DNA. Not very different, okay? But it has some different shapes. And because it has different shapes, it has different jobs in the cell. So we're going to learn about 3 RNAs. Are you ready for them? The first one is messenger RNA, which we abbreviate M. So if you want to, let's draw a little arrow from messenger to M. Okay, so when we say mRNA, that stands for messenger RNA. And you can see a nice picture of it, right? I would describe this one as linear. Kind of looks like a straight line, right? It's a polymer. It's linear. And its job is to make a copy of a gene and carry that copy to the ribosome. We're going to think of this as kind of like a letter. It's kind of like a letter. DNA is the author of the letter. The letter is the messenger RNA. Any questions so far? How's my pace? Too fast? Ribosome is the next one. The R in ribosome is the R that we're abbreviating by saying rRNA. It's not a typo. There's 2 R's there. The first one's for ribosome. The second R is for ribonucleic acid. So rRNA, this guy I think of as kind of like a hamburger. Can you imagine this as a hamburger bun? The big bun is on top, and the bottom bun is at the bottom, right? The smaller bun, no one sees that one, right? So it can be small. You want a big, plump, sesame seed coated bun on top of your hamburger. So there's 2 parts to the ribosome. And this is going to be kind of like a mailbox. It's going to receive the messenger RNA. So the messenger RNA is a letter that's written by the DNA. That letter travels to the mailbox, the ribosome, and then the transfer RNA, or tRNA, is going to read that letter. So we're going to think of the tRNA as the reader, okay, or the translator. Now this tRNA is the most interesting one. I think it kind of looks like a clover leaf, maybe an upside down clover. clover shape. Some people say it looks a little bit like a lowercase t, which is helpful, right? tRNA looks like a lowercase t. There's a couple of key features, though, we need to highlight here. This is what's called, these three special colored ones are what are called anti-codon. Anti-codon. Anti is a prefix that means against or opposite. So the anti-codon is the opposite of the codon. Well, what's a codon? In order to talk about that, we need to go back over here to messenger RNA and circle the first three nucleotides. The first three nucleotides on the messenger RNA are called a codon. Okay, so we have a codon on the messenger RNA, and we have an anti-codon on the tRNA. One more thing that we have to add on here is we're going to add a little ball on the other end of the tRNA, and we're going to label that ball an amino acid. An amino acid, remember, that's the monomer for a protein. Man, so much to label. We're not even halfway through the first page. Anyone feeling anxious? Take a deep breath. Hold it. Let it out slowly.
Mr. Kramer, speaking of the tRNA, it does look like a lowercase t except for this bar.
Yes, I think that's the cross on the T.
Yeah, the cross.
I think that's the cross on the T. We could have said this looks like a cross. Kind of looks like a cross.
Yeah, but
Green looks like a cross.
But the horizontal line's a little crooked.
It is a little crooked. It's got that thing sticking out there on the side.
Looks like a pimple.
Yeah, a little bit of a pimple in that. Don't forget we ended for 33. 33 today. Holy cow, and we got to hold a snake for at least an hour. Anybody need more time with this? We've got 3 different kinds of RNA. I see it's 3 different kinds of RNAs. And these RNAs are going to be building the proteins. Okay, the reason we're talking about these 3 molecules is they're going to build our proteins. They're going to make the tools using the information from the DNA. You good, Samantha? Awesome. Anybody else? Any more time? You good, Isabella? All right. Here we go. All right. Is there anything we need to write down for this? Let's take a look. Protein synthesis, making proteins. Here's what we're going to do. Think of this as like a workflow or a flow chart here, okay? We begin here. Let's write begin on this side. We start with the DNA that we got from mom and dad, right? And that DNA is going to get copied into a messenger RNA. And the process of making that copy is the first step of protein synthesis, which we call transcription. Transcription is the first step. And when you look at transcription, don't focus on the trans part, focus on the script part. When you script something, it's about writing, right? So transcription means to write something, right? In this case, the DNA molecule is writing a copy of itself, which is the mRNA. Which remember, the mRNA stands for messenger RNA, and it has little words that we call codons. We call codons words? We didn't make that reference, did we? The codons are words, right? Just like I'm speaking words to you right now, these are words. The second step is translation. That messenger RNA has to get translated. You guys probably know about translation, right? Many of you are taking a foreign language, or not a foreign language. Foreign languages don't exist anymore. They're world languages. Worst teacher ever.
And also, I know who can speak about many forms of communication.
You can?
I said I know somebody who can.
Who can do it?
probably knows me.
Oh, okay. Who?
From Star Wars C3PO.
C3PO, right?
So he can speak 7 million languages now. He upgraded his language.
That guy is amazing, right? And he's so good with the statistics. That's R2D2. He just translates R2D2. R2D2 is our statistics robot. Yes, we need them here now. Nucleic acid, that's a language. We talked about this before, right? The order of those nucleotides matter. It's a language, and that's going to get converted into proteins, which is a different language because there are different monomers. There's different monomers in nucleic acids. So basically, this is where we convert from the nucleic acid language, nucleic acid code, into a protein language. And that protein language, of course, is what makes that protein have its very special shape. Okay, Kim. Yeah, that's great. Yes. Cam's asking about the mRNA that was used in the COVID vaccine, right? Yes. And what did you learn?
I don't know.
Yeah. Yes. So these mRNAs, nowadays, it used to be that we used to inject viruses into people that have been weakened, and that would allow their bodies to fight them off and develop immunities. Well, it's a little bit dangerous with a very infectious virus, right? We throw a weakened version in, it might still win, kill the person. So the idea is, well, what if we don't put the virus in, but we put in the instructions to make the virus? A little bit safer, right? So we put in the instructions to make the virus, that's what's in the shot, and then our body makes the virus, but as it's doing it, our immune system has a head start. Yeah. Right. That's a little bit different, though. They're using a virus to deliver the new genetic information. So we've also done genetically modified viruses where we take out all their bad DNA that makes them hurt us, and we put in DNA that we want to give to an organism, and then we just infect them with the virus. So it delivers all of the helpful DNA that we want to modify. Boy, this is good. You guys are asking questions. It's great. Any other ones? This is very relevant. Yeah, this is good. I was wondering how I could make this relevant. You guys provided the relevancy. That's awesome. So again, we're going to think of messenger RNA as kind of like a letter. The ribosome is like a mailbox. And the people reading the letter are kind of like the tRNA. So let's continue on. Does anybody need more time? Samantha, you good? Brandon, you good?
Yeah.
Okay. Here we go. Here's a little map. Okay, where's all this happening in the cell? There's not a lot to write here. I think maybe what we'll do is let's just circle the word transcription, right? And let's underline the word DNA. It all starts with the DNA that we get from mom and dad. Transcription happens, and we make messenger RNA. Exactly. You got it. It's just a shorter, think of the messenger RNA as a short version of the DNA. You got it. Exactly. That's exactly it. And of course, this is the nucleus where this is happening. So transcription, the first step, happens in the nucleus. Then notice the messenger RNA leaves the nucleus, and we go to the second step. There's our ribosome, which if you want to, you could abbreviate rRNA, right? This, of course, is our messenger RNA. So the ribosome covers over the messenger RNA, kind of like a hamburger inside of a hamburger bun, okay? So think of the messenger RNA as kind of like a hamburger, although I'd say it looks more like a hot dog. Can you imagine a hot dog and a hamburger bun? That's kind of what we're looking at here. The messenger RNA is long like a hot dog, but the hamburger bun sandwiches that messenger RNA. They're trying to represent that with this like shaded thing here. And then this is our tRNA. Remember the tRNA has the anticodon and the amino acid. Let's label that. So there's the anticodon. And Jenna, I know what you're thinking. That looks even less like a cross, right? And that little ball there would represent.
Yeah, it looks more like an outlet.
These are artists.
Wow.
Artists are doing this, right? And then let's label here. There's the codon. We're going to think of codons as like words.
Yeah,
and all of this part is happening where? It's happening wherever you find ribosomes. So remember we said ribosomes can be in two places in the cell. They can be just floating around like this one is, or they can be found stuck to the endoplasmic reticulum, or the ER. Any questions here? How are we doing, Tom?
I do. The tRNA looks like an outlet.
Like an outlet. Yeah, a little bit.
Yeah, an outlet plug.
With the ground and the two prongs, yeah. Anybody need more time? We're going to think of this as kind of our map. Okay, this is the map where the information goes from, oh, one more thing I wanted to label. Sorry. This is kind of the whole point. Circle this thing. That there represents our protein. That's our tool. Right? There's the tool. That's the protein. That's going to fold up, just like a pipe cleaner folds into a very special tool shape. Maybe it's going to be a hammer. Maybe a chainsaw. But its shape is going to allow it to do its very special job. Any questions here? All right, here we go. So now we got to get into the nitty gritty here, okay? We're going to talk about those two steps, transcription and translation. There's two steps that got to happen, but I think the first one is super easy because we've already talked about something that's very similar to this, right? Look at the steps here. The DNA unzips. Do you remember that? We talked about this before, right? It's when DNA got replicated, right? So it's the same thing. The DNA unzips, that breaks the hydrogen bonds, and then look at step 2, free-floating nucleotides come in and base pair. So let's put a box around the word unzips. We're going to call that the unzip step. First, the DNA unzips. And then let's, on the second step, let's box in base pair. They base pair. We know what that means, right? G with C, C with G, T with A with... here's a little difference. Right, it's supposed to be A with T, but the problem is in the world of RNA, there's no such thing as a T. And so apples in the tree doesn't work anymore. I like to remember this as Army uniform. Why? Because it works. A is going to base pair with a different nucleotide called uracil, which we'll abbreviate with a U. Okay? So notice on the DNA side, there is a thymine, but on the RNA side, there's no thymine. It's just uracil. Okay, so just have to remember that. It's a little bit tricky. Cards in the garage, yes. Army uniform when it comes to pairing up with that. Okay? Step 3, RNA polymerase bonds the mRNA together. That should sound familiar, right? We talked about DNA polymerase before. This is just a different tool. It's just a different enzyme. We're going to think of it as a stapler. It's a stapler tool. RNA polymerase. Yeah, RNA polymerase, that's the next tool. Then the DNA rezips, goes back to being DNA again, and the mRNA floats away. Let's just underline or box in mRNA. And so this is how we get an mRNA from a DNA. Number 1, the DNA unzips, that breaks those weak hydrogen bonds. Number 2, free-floating nucleotides come in and base pair, G with C, C with G, T with A, but A with U. RNA polymerase comes along and staples them together. The DNA rewinds or rezips, and the mRNA floats away. And so, as Owen was saying before, mRNA is just a little copy of a section of DNA. Say again? No, it doesn't. It doesn't. It just means that not all of the DNA gets translated all the time. No, and actually, if it did do that all the time, you would have a condition like cancer, right? Where we're creating genes that we're not supposed to be creating or expressing and creating all kinds of problems. Yeah. So it's important that only the right genes get turned on at the right time. We don't want a free-for-all where everything's being turned on at the same time. Yeah, great question. Any questions on transcription? It's a lot like replication, except at the end of this, we don't end up with two DNAs. We end up with a messenger RNA. Okay. Anybody need more time? How we doing? Samantha, you good? Brandon, you good?
Yes, it's a good free shot.
Okay. Lindsay, you good? Okay. How are you today, Lindsay? Good. I'm glad you're here. Okay, so let's do a little practice here. This is something that you'll be asked to do lots and lots. What we have to do is, here's our DNA strand, TACGCGACT, and our messenger RNA is going to be the complementary copy of it. So what is going to come in and stick to a T? Say again? U, close? A. T is still going to stick to A, okay? But here's the new one. If there's an A on the DNA, then what are we going to stick to? That's the U. Okay, so T goes with A. You remember, DNA can still have T. It's just RNA that doesn't have a T. So RNA's A will stick to T. RNA's U will stick to A. What's going to stick to a C?
G.
G, same as always. Somebody raising their hand and I'm ignoring them. Who is
Oh, that was me.
Oh, Jenna, you never.
I was just gonna say G.
Yes, what about the next one?
C.
You're right. G with C. Great job raising your hand. C is going to stick with G. How about G is going to stick with a C? Great. Uh-oh, and there's another A. U. Remember, Army uniform. C is going to stick with a G, and T is going to stick with a A. Okay? And so that's the complement of the gene, right? The DNA up here, that's part of the gene, that's part of the DNA that's in the nucleus. The messenger RNA is just a little bit of that DNA. Go ahead. Right. T with A with U, when we talk about RNA. So it's a little bit tricky to remember. Okay, we're motoring. Step 2. Remember, we said now we've got a messenger RNA copy of a DNA gene, and so now we've got to read that message, right? We have to translate it. We have to convert English into Spanish, right? In this case, we're going from nucleic acid into protein. So we want the same meaning, but we want it to be a different sequence of monomers. And so the steps to translation: ribosome attaches to the mRNA at the start codon. So let's just box in the words start codon. This is the start codon step. Remember, a codon we can think of as a word. In DNA's world, the codons are always 3-letter words. They're always 3 letters, 3 nucleotides. And the start codon, the first word of every DNA sequence, is always AUG. So that might be one that you want to memorize. You don't need to learn the DNA language, but there is one that would be helpful to know off the top of your head, and that's AUG. That's always the first word in the DNA sequences.
In fact, AUG is abbreviation for the month of August.
Oh, you're right. I've been teaching this for years, Jenna, and I never made that connection. It's August. So just remember, August, AUG. Okay. Somebody raise your hand.
Sorry, that was me.
That was it, the false alarm. Okay. All right. The tRNA is going to bring the correct amino acid for each codon in the ribosome. So the next step is going to be the tRNA step. tRNA is going to bring the amino acids. So maybe we need to label that. This pink ball, remember, that's an amino acid. It's a monomer for a protein. Look at this. This looks even less like a cross. Our drawings are getting simpler and simpler. There's lots of different ways that artists try to represent these things. But remember, the tRNA has an anticodon that complements the codon. And you can see that, right? Take a look right here. There's a codon, AAA, right? Look at the anticodon, UUU, right? A sticks with U. You see how they fit together? They're opposites of each other, right? And so UUU is the anticodon. That's always with the amino acid lysine. And so we're forming bonds between these amino acids. We're making a protein. Remember, that's our tool. That's what's going to allow our cells to function. And so then the ribosome moves to the next codon and brings the next amino acid. So I think of this as kind of a repeat, right? Repeat step one, do step two, repeat step one, go to step two. And just keep repeating until you build this long protein. Any questions here? That's what translation is. And so this is how we can go from messenger RNA, our nucleic acid, using a ribosome and tRNAs, we can make a protein. Did you know this is happening inside of you? It's happening right now. Ella, what's messenger RNA?
No, I can't do it.
tRNA. Anticodon? Amino acid. Oh, in it. Oh, I'm just drawing lines to show that A sticks to U, A sticks to U, A sticks to U, that they fit together, that they complement each other. Yes? Yes, that's a great question. This is, in biology, they actually have a name for this. They call this the central dogma, which famously was, I think, first used by Crick, Francis Crick, who was one of the co-discoverers of the DNA double helix. But yeah, this is happening in bacteria. This is happening in dogs and in trees. And this is why I can take a human gene and put it in a bacteria, and the bacteria knows what to do with it, right? I mean, it's all the same equipment, all the same machinery. All right, well, let me show you how we as humans can translate. Our molecules know how to do this without us, which is really cool. But we need to know how to do this as well. We've got a chart that helps us do that. Does anybody need more time underlining and circling and things? All right. So here's our last little bit. Is this, yeah, we're not going to do this last page. We'll do this, this is where we'll stop for today, okay? So here we go. Let's take a look at this. Now we've got messenger RNA, AUG-CGC-UGA, right? And let's go ahead and label. Can you guys label codon? Remember, codons are 3 letters. One thing that's a little bit weird about DNA is it doesn't put any spaces between the words. So the words just kind of run together. And so you have to remember, every 3 letters, that's a codon. And so the next codon would be the next 3 letters. Maybe I'll put codon number 2. And then the next 3 letters are going to be codon number 3. No spaces between the words. It's kind of annoying, right? But that's how it is. Save space, right? So how can we as humans interpret DNA into amino acids? Well, we have these charts, okay? And I'm going to recommend that either one's good. Either one's good. But let's look at this one on the right first, this circle chart. I think this one's kind of the easiest to work with. You see how there's 3, well, there's a bunch of rings, actually. But if you look here, let's put the number 1 on the first ring, on the innermost ring, that yellow ring. Then let's put the number 2 on the next ring out from the middle. Then let's put the number 3 on this reddish ring. And then where it's light blue, let's put AA for amino acid. If you don't think you'll remember what AA is, maybe we should write it down. Amino acid. Okay, and so the way this works is if my 3 letters are A, U, G, then I'm going to look for A in the first circle, U in the second circle, G in the third circle, and then the protein or the amino acid in the fourth. Okay, so let's look here. AUG, that's going to be A here, U here, G here. And so the amino acid for that first word is methionine. Now, I don't want to write that because I'm lazy. So there's an abbreviation in the last circle that I'm going to use. MET. What this word? Amino acid, sorry. I abbreviated it AA. Amino acid. You see how I did that? AUG, first word, first letter A, second letter U, third letter G. That meant it was this amino acid, but I didn't want to write the whole word out, so I put MET. Okay? Let's do the next one. CGC. So in the first, or the innermost circle here, I'm going to circle my C. C, then what? G, there's my G. And then C, that makes arginine. Arginine is a different amino acid. I don't want to write it out. I'm going to use the abbreviation ARG. You see how this chart works? Let's let you guys do the last one. See if you can do this last one on your own. Start with the U, then go to the G, then go to the A. You see the word there? Or do you see it? Start with the U, then go to the G, then go to the A. Stop! Which is what Isabel's been trying to get me to do this whole time. Just stop. So we'll write in the word stop. What do you think stop means? Is there an amino acid named stop? That's a weird name. No, it just means it's over, right? It just means class is over. It just means that the protein synthesis is done. It's like a telegram. Yep. Yep. And this is how we can interpret the telegram. Now let me show you. If maybe you don't like circles, maybe you like squares and rectangles. You can do this as well. So let's look at the left side. Put a 1 on the left side, put a 2 on the top side, put a 3 on the right side. So the way this one works, AUG is the first word, right? So if I look at A and U, I know it's one of these ones. And then if I look over here, I can find G. It tells me that that first one is methionine. Different way to get the same information. I like the circle a little bit better, but it's up to you. Let's look at the second one. CGC was the second one, right? So if I want to find, oh my goodness, that was supposed to be an eraser. If I want to find CGC on the first side, left side, C and G, I know it's going to be arginine. I don't even need to look at the third one, but I could. It's that. So ARG. So again, whatever you want to use, squares or circles, doesn't matter. All right, well, let's go ahead and upload these notes to Canvas and break out a snake. Thanks, guys.